Today we are continuing talking about fungi, and specifically about their cultivation and laboratory investigation. Last week we discussed about the fact that as well as we know, fungal infections are not the mostly spreaded ones. But still, it's a very big issue in a healthcare system in any country, for a simple reason. First of all, most of them are making part of normal flora of our environment. Second, there are a lot of them which are mandatory pathogenic bacteria, mandatory pathogenic fungi. And even if it is not a pathogen, it could be an allergen. So if in your place there are not good enough conditions, there is a high level of humidity, there are some hygienic issues in people's houses, it becomes a big problem. If I would ask you to tell me, do you know a person with allergy? Do you know any person with allergy? Yes. Who doesn't know any person with allergy, please? Who doesn't have any allergy? A little bit more. Few years ago, I would raise a hand with you. So I thought I don't have any allergy. The reality is that any organic substance can be an allergen for people. So even if we are talking about non-pathogenic fungi, even these substances or these organisms, macro or microorganisms, are presenting high risks. Some relatives of ours are thinking, some relatives of mine are thinking to move, to change their place just because they are not able to fight in their place with some fungi, and the child has allergy. So they literally are thinking about changing their living space. So it is a big issue. If we are talking about lab investigation for fungal infections or for presence of fungi, we can divide them in different groups. Of course, there is nothing that is cut out from or taken from the context of clinical evolution. So very often fungal infection can be diagnosed simply just by clinical investigation. What means clinical investigation? When you appreciate symptoms. Clinical investigation is when you look, it is called, do you remember? Inspection. When you just look at the patient. It can be, if we are not talking about infections, if we are talking about general clinical investigation. It can be inspection, palpation, auscultation, percussion. Do you remember it? Do you know it? So you look, you touch, maybe you... Percussion is when you... How you call it? Not hitting, but percussion. Palpation, auscultation with a stethoscope, or maybe without stethoscope, but you're listening. And these clinical features, also you are talking to a patient, you are asking for how long, for what does bother you, what's going on, what's wrong, and so on. It is clinical investigation. So it is a contact with a patient. So you will observe, if you will discuss with the patient with a fungal infection, probably it will be a contact infection. The variety is very wide. Some people will tell you they are staying maybe in dorms, right? And they have a common shower room. And some of them maybe don't use their own their own, how we call it, shoes for showering shoes, slabs, or slippers, yes. So and they are just walking like that without anything, and after that some itching in the top skin, yes. Could it happen? Or you are coming in the room and you don't wear shoes in the room, and your colleague does, and it can happen. Or it's summer and you're walking around with slippers, yes. Outside there is dust outside. This is how anyone can get a fungal infection of the planetary skin, yes? Some itching, some maybe changings on the skin, some rashes, and so on. This is the way how you can get skin infection, bacterial infection. This is how you can get fungal infection. This is how you can get parasitic invasion, and so on. Direct contact. Especially if there are some skin lesions, right? If there are some skin lesions, there is a higher chance to get an infection, fungal, bacterial, viral, whatever. With the skin lesion, we have extra gate for entrance of the pathogen. So in most of cases, the way of infection will be direct or indirect contact. Which is the difference between direct contact and indirect contact? When we are talking about contact, we are talking about contact with what or with whom? With the source of infection. And what is source of infection? What is source of infection? Huh? In some cases, it is a human. In some cases, it is an animal. And in some cases, it is kind of environmental factors if we are talking about sapronotic infection. Sapronotic means from the decayed rotten material in the environment, not from a living body. So if you have a direct contact with the source of infection, it will be direct contact infection. What means direct contact? Handshake. Any skin infection can be obtained through any kind of skin-to-skin contact, starting with handshake and finishing with kisses and sexual contact. Yes? Yes. This is the way how direct contact infections are obtained. Indirect contact, when you are touching same items or same surfaces, same things, the source of infection have been in contact with. So, source of infection have been touching this phone, I am touching this phone, there is a chance for me to get infected. The source of infection, human, have been touching this surface, I am touching it, there is a chance. Especially if I find some skin lesions on myself. And not that often, but still, through inhalation, if it is a dust that could contain some spores of fungi, there is a chance to get a respiratory tract infection caused by fungi. And if there is extremely bad situation, for example in hospitals, with disinfection, with sterilization, with it basically doesn't happen too often, almost impossible, but still some nosocomial infections can be caused, including by fungi. So after clinical evaluation, we need to make a specimen collection. And here it is important to pay attention which is the reason why we take this specimen. By reason, I mean which is the target or which is the method of investigation you will apply. Because for some methods, it is very important to take it patiently, to not damage it from the moment you take it till the moment you will make investigation. It is specifically about microscopical investigation and about cultivation of fungi. But if for example you take it for some biochemical investigations or for some molecular biological investigation, like PCR, like Malditov, like these tests, you don't really care about will this pathogen die till the moment you make investigation or not, because you are looking just for some samples of genetical material or for some samples of some pieces of the pathogen. Also, if we're talking about antigen-antibody tests, you also don't really care, because even if it will die, even if you will even if the sample will not survive till the moment we will start investigation, there are still some samples of antigens that will enter in the reaction with antibodies and will have positive or negative reaction. This is if we are talking about antibody tests. And this is what is universal about antibody tests, serological investigation. It is universal for anything. It is universal for for bacterial infections, for viral infections, for fungal infection, for any kind of infection. Antigen antibody test or serological investigation will be universal and will be extremely fast, will be very affordable, will not require too much facilities for this examination. Also, histopathological examination. When we are talking about histopathological examination, it is either we are talking about tissues we take from patient during the life, it is biopsy. Either we are talking about specimens we take post-mortem, after death, it is called autopsy, right? By the way, does anyone know the only 100% investigation method? The only method of lab investigation, or any method or any diagnostic method in medicine that gives 100% result all the time. And it is the mostly useless method at the same time. The mostly precise and the mostly useless for patient method. Any ideas? Any ideas? Which is the mostly precise way of investigation in medicine that is at the same time the mostly useless for patient? Huh? No? No any ideas? Meanwhile, I gave you a clue. Autopsy, yes. Even if during the treatment you made a bunch of investigations and some of them wasn't precise enough, after death you have time for investigations as much as you need. And finally you will find the truth. But who cares? Okay, so, but still it is the mostly precise method. So if we are making histopathological examination, we can see not only the pathogen, but also those morphological changes that will happen in tissues during this infection. And why it is that specific for fungal infections? Because they are giving mostly a specific picture. If you will observe, if you will make comparison with bacterial cultivation and fungal cultivation, you will observe that one of the mostly beautiful results is giving fungal investigation, right? There are some doctors who are making pictures with on culture media by using different types of fungi and bacteria. So also in tissues they can produce some colonies and some pictures that if it wouldn't be that sad, it would be awesome. Yes? And in dependence on what we want to see or which result we are looking for, we have different methods of cultivation of fungi. What is in common between cultivation of fungi and cultivation of bacteria? In both cases, we will use culture media. It is not in common with viral infections. For viral infection, it doesn't make any sense to use culture media, but for fungal infections and bacterial infections, we are using culture media. It can be solid culture media, it can be liquid culture media, it can be on a Petri plate or in tube. Different types of culture media we can use, we can apply for investigation of fungal infections. But specifically it depends on our targets we have. Some of them are important specifically just for isolating some infections. So it is the mostly simple way of multiplying, just as in culture media, you know. Some of them are simple culture media just for cultivation of some specimens, of some pathogens. Some of them are used in order to applied in order to obtain either the fungal mass, either the results of fungal metabolism, because fungi are applied in industry for different products, for production of penicillin, antibiotics, for some enzymes, for some maybe for some proteins obtaining, or some toxins. So when we need to produce some enzymes or bioactive compounds, we can make liquid culture. So it will be much easier to separate the solid side from a liquid side, and from a liquid part you will obtain all the materials, all the metabolic results you need from this fungi. Also, there are ways of cultivation of fungi used just in order to, like making a storage of samples. Because if you need to obtain a sample, and you need to have it for a long time, you need a compact storage. So some culture media, some methods of cultivation are used simply just to make storage of samples of fungi, and this is how it will look. They will be applied in slant cultures. Also, we have deep fungal cultures. This is how it looks. So, these culture media are applied in order to see the behavior of fungi. So here is an example of potato dextrose agar. So if you want to see how it grows, how it manifests, how it looks, and which is the behavior of some samples in nature, you apply this kind of culture media. Here is an example of growing of Aspergillus niger. This one is basically applied for investigation in the field of ecology. So you just make investigations how fungi are manifesting themselves in nature. Also, it is very interesting way of cultivation, and probably, I guess, look how it looks. Bioreactors, how it looks like. The device, device looks like anaerostate, right? Do you remember how it looks? Is a jar with a pump with when you put their culture media and you pump out all the air. This is how it looks. Looks similar, but in these bioreactors, we introduce their specific culture media. So bioreactor is connected to different suppliers. That gives opportunity to supply all the time with necessary liquids, nutrients, and so on, in order to cultivate some specific types of fungi. And this method is provided specifically created specifically to provide as much as possible biomass of fungi. If we need to obtain as much as possible actually the mass of fungi, we will supply constantly the bioreactor with all the necessary nutrients, and that amount we need, that amount will grow, will multiply. Also, solid state fermentation method of cultivation of fungi, also a compact and specific one. So this method is applied also for obtaining of specific materials provided by fungi, including obtaining some organic substances, some acids, some fats, some proteins, some biologically active units or some biologically active substances, also obtaining of microbial biomass. So some fungi are living together with bacteria, and it is called how this form is called when they have mutualism, when they have mutual benefits. Mutualism, and one more synonym. Huh? Louder? Cooperation. Okay, it is more for humans. When two different species are living together and producing benefits to each other. Symbiotic, yes, symbiotic life. Thank you. So some fungi can be used for to help to provide production of microbial biomass as a kind of symbiotic microorganism. Also, such kind of investigation method, suspension cultures. Some cultures are growing inside of liquids. Some cultures are growing on the surface. So this is why it will be called suspension cultures, because they will be suspended on the surface of liquid of the culture media, and will produce also a fungal biomass or product. And also if you need the biomass, you collect it from the surface. If you need the products of metabolism of the fungi, you collect it from liquids. So it is separated already from the one from another one. So this method is highly used also in biotechnology processes, in pharmaceutical industry, for production, for obtaining of specific biologically active matters, and also in alimentary industry and environmental industry.